Hi and welcome back to Pharmacology of the Synapse. In this video, we're going to go over the mechanisms of actions of the benzodiazepines and look at the GABA-A receptor and its chloride channel. So sedative hypnotic drugs act on the GABA neurotransmitter system, and GABA is the primary inhibitory neurotransmitter in the brain. GABA acts on GABA-A receptors, which are chloride channels, as well as GABA-B receptors, which are GPCR-type receptors. In these videos, we will be discussing GABA-A receptors only. Activation of GABA-A receptors results in an influx of chloride ions into the cell, which hyperpolarizes the neurons, making it harder for the cell to depolarize and fire an action potential. This ultimately decreases the excitability of the neuron. Now, GABA-A receptors are formed from 5 different subunits. There are GABA binding sites on each of the alpha subunits, here in blue, and then benzodiazepine binding sites are on a different subunit, the gamma subunit. Benzodiazepines act on GABA-A and not GABA-B receptors because those are different receptors altogether, and GABA-B receptors do not have benzodiazepine binding sites. Both benzodiazepines and barbiturates act as positive allosteric modulators of GABA-A receptors, meaning they enhance the function of GABA and can't do anything on their own. Benzodiazepines and barbiturates potentiate GABA's inhibitory effects on CNS function. Some of the color coding that appeared in the previous slide can be found here. GABA-A receptors have a rich pharmacology with lots of different binding sites and effects. You have GABA itself, which binds at the endogenous binding site, and GABA is the inhibitory neurotransmitter. There is a GABA antagonist, bicuculline, which is an antagonist at that same GABA site. Now, bicuculline can produce seizures, which makes sense if you think of antagonizing an inhibitory process. This can increase excitatory processes, which can result in overexcitation and seizures. The benzodiazepine binding site is the site of action of the benzodiazepines, such as diazepam, and these drugs potentiate GABA function. There is also a benzodiazepine antagonist, flumazenil, which can antagonize the action of benzodiazepines only, not GABA itself. And lastly, there is a benzodiazepine inverse agonist, beta-carbolines, that can produce increases in anxiety and aggression, so kind of opposite to the anxiolytic and sedative properties of benzos. There is also the barbiturate binding site, an ethanol binding site, and lastly, you can block the entire chloride channel with picotoxin itself. Here you can see a cartoon of the GABA-A receptor with the 5 subunits surrounding this central chloride channel. GABA binds to one subunit, and when GABA binds, it opens the chloride channel and you get chloride influx into the cell. Now, the benzodiazepines bind on a different binding site and they can potentiate GABA function, so you get a larger influx of chloride into the cell. Remember, benzodiazepines cannot activate the GABA receptor and cannot open the channel on their own. To further illustrate this benzodiazepine potentiation, we can turn to some electrophysiology, where we insert an electrode into a cell and can record the electrical activity in the cell. On the left here, there is a whole cell recording, and the step function describes when GABA is applied to the cell. You can see the downward deflection of the current trace when GABA is applied. When the benzodiazepine flurazepam is present, you'll note that nothing happens until GABA is applied, but once it is applied, there is a larger downward deflection in the current trace. On the right side, you can see synaptic current traces that result from single vesicles of GABA being released into the synapse, and when they bind to the postsynaptic side, they evoke a small picoamp current. You can see that the frequency of these mini inhibitory postsynaptic currents, or mini IPSCs, is unchanged, but the amplitude of them is bigger when flurazepam is applied, reflecting the potentiation of GABA by the benzodiazepine. Another way to conceptualize this is that GABA itself evokes a certain size of chloride current, so here 100% of the GABA current. When you add benzodiazepines, you potentiate this GABA current. If you block the benzodiazepine with a benzoantagonist, it doesn't affect GABA's ability to produce a chloride current, you just block the benzodiazepine potentiation of the current. However, if you block the GABA binding site itself with an antagonist, you can reduce the chloride current, and the benzodiazepine will no longer have any potentiation effect because it has to have GABA present to work. GABA is released from inhibitory neurons, and the synapses where GABA is released from are often called inhibitory synapses. Here you can see a figure 5.7 from the textbook that illustrates a GABA synapse. GABA is synthesized from glutamate by glutamic acid decarboxylase, or GAD. And GAD is often a marker of GABA neurons because you need this enzyme to produce GABA. GABA is then packaged into vesicles by the vesicular GABA transporter, or VGAT. Note that VGAT is also able to package another inhibitory neurotransmitter, glycine, into vesicles. I've put the GABA A receptor here on the postsynaptic side that would then receive the GABA inhibitory signal from this presynaptic neuron. GABA neurons are often inhibitory interneurons that act to dampen neural circuit activity. The schematic here shows two neurons where the one on the left excites the one on the right, following down this green arrow. A GABA inhibitory neuron would act on the left interneuron. The GABA inhibitory interneuron would then act on the left neuron in releasing GABA and inhibiting this neuron, which then would ultimately reduce activity on the right neuron as well, dampening down this entire circuit.